I typically like to start our genetics unit with something called PTC paper. So with PTC paper, I give a piece to every student, and it's a tiny piece of paper that you put on your tongue. And immediately, when students put it on their tongue, you see a variety of reactions around the room. Some students are repulsed by the taste of the paper. They think it tastes absolutely terrible. Other students are kind of indifferent, like they can tell that it has a strange taste, but they're not totally grossed out. And then some students cannot taste anything. So that is because PTC paper has a chemical on it, and some people are genetically predisposed to being tasters, and then other people are not. So they've studied this PTC gene, they call it, and people who are not tasters tend to be homozygous recessive for that trait, which means they have to have two recessive alleles, one from mom and one from dad, and this is typically not as common. So usually you'll have a lower number of people who cannot taste the PTC paper. I would say about 75% of people can usually taste it. And if you're a taster, like I'm a taster, I think it tastes really, really bad. Um, you're either heterozygous or homozygous dominant, which means you have the dominant allele and from at least one of your parents. And so hopefully you get to do it someday. Stop by my room next year and I'll give you a piece of PTC paper and you can try it out. So we are going to talk about Mendellian genetics first. We're going to get into monohybrid crosses, and then I'm going to briefly introduce you to dihybrid crosses. We need to break down a few terms before we get into Gregor Mendel. The first term is heredity. And heredity is the process in which organisms inherit traits from their parents. Genetics is the study of the patterns of inheritance and variation. So genetics is just the study of heredity. So Mendel is such a cool dude because he was the father of genetics. He crossed pea plants to study the inheritance of traits. So Mendel discovered a few very important things that geneticists and scientists still use today to further and advance genetic research. In the late 1880s, Mendel experimented with a little over 30,000 pea plants. So through his experiments, he determined that physical traits are determined by the inheritance of genes, and genes are passed on from parents to offspring. So he also figured out that the dominant gene from each parent determines the physical trait that is observed in the offspring. And this was huge. This was a huge discovery. So let's first talk about why he used pea plants. So Mendel used pea plants because they were very easy to grow. You could also track one generation to the next in a short period of time. If we were to do this with human beings, our lifespans are much longer than a pea plant. So he used pea plants to simplify this process. He also used pea plants because they can reproduce sexually, so they have gametes, and he was able to cross-pollinate them due to these gametes to produce and assess variety and where this variety was coming from. And then lastly, pea plants have very distinguishable traits. And I'll show you some of the things that he studied. He studied flower color, pea shape, pod shape. It was incredible, everything that he breaks down and, not only gathered data, but brought meaning to the data. And another cool thing about Mendel is he practiced good science because he used such a huge sample size to make sure he was getting the same results each time and that his experiment was valid and reliable. Mendel first crossed purebred pea plants. And what he found was a cross between two purebred pea plants produced the same color flower. So if you crossed two purple flower plants, you get purple flowers. You cross two white flower plants that were purebred, you get a white flower result. And so that was his first experiment. Then he went on, and it got a little bit more complex. So first thing I want to mention, the P generation is the parent generation. Their offspring produces the F1 generation, and then their offspring produces the F2 generation. So he purebred white and purple pea plants, and then he crossed these two variations. So he crossed a purple plant with a white plant, and when he did that, two purple plants were produced. He got two purple flower plants. And this was puzzling because it was like the white gene had disappeared. It was like the white trait has totally disappeared. But then he went on and let the F1 plants self-pollinate. That created the F2 generation, and the white flower reappeared in the F2 generation. So this led Mendel to believe that there are two different alleles for each trait, and an allele is different forms of a gene. So there are dominant traits, there are recessive traits, and sometimes in certain generations, the recessive trait might be masked by the dominant allele, but it could show up in a later generation, depending on the combination. And so that was his major discovery from cross-pollinating the pea plants. So Mendel not only did this with flower color, but he discovered dominant and recessive traits for plant height, seed color, seed shape, pod color, pod shape, and flower position. So he tested many different traits to confirm his results.
After observing the results of his first set of experiments, Mendel wondered whether different characteristics are inherited together. For example, are purple flowers and tall stems always inherited together, or do these two characteristics show up in different combinations in offspring? So to answer these questions, Mendel next investigated two characteristics at a time. For example, he crossed plants with yellow round seeds and plants with green wrinkled seeds. The results of this cross, which is a dihybrid cross, are shown in the figure to the left. So Mendel repeated this experiment with other combinations, like flower color and stem length. Each time, the results were the same as those in the figure. The results of Mendel's second set of experiments led to the discovery of the second law, which is the law of independent assortment. It states that factors controlling different characteristics are inherited independently of each other.
Mendel concluded two major things. The first was inheritance is determined by factors that are passed from one generation to the next. And two, some alleles are dominant and others are recessive. So this brought him to discover two laws. The first one's called the law of segregation. That's when allele pairs separate during sex cell formation and meiosis and unite randomly during fertilization. And then the law of independent assortment, and that is each trait is inherited independently of each other because of the random distribution of alleles that occurs during meiosis. So that kind of circles us back to the topic of gamete formation and meiosis. I also want to review the vocab genotype and phenotype. You were exposed to this in life science in middle school, but I know it's been a while, so we're going to circle back. The genotype describes the set of allele pairs present in an organism. If both of the alleles or trait are the same, we call that homozygous for the trait. If the alleles for the trait are different, we call that heterozygous. So the genotype is the gene combination of the organism. So we represent that with letters. It's the gene code. Phenotype, on the other hand, that's the way that the organism looks. So it's based upon its physical appearance. You can remember this because phenotype, physical. They both start with pH. So in the example of pea plants, the plant with purple flowers had a phenotype for purple flowers. The plant might have the recessive gene for white flowers, but this doesn't matter in its phenotype because of the dominance. The recessive gene is not always expressed, as we discussed on the last slide. So your phenotype is how you look. Like, are you tall? Are you short? Do you have red hair? Do you have brown hair? Do you have blue eyes? The genotype is your actual gene combination, your code, your genetic code. So this principle of dominance is saying that if the dominant trait is present, then it will be expressed in the phenotype. So the dominant trait is the trait that is expressed. It appears if the trait is there. So this is represented with an uppercase letter in a genotype. The recessive trait, on the other hand, that's the trait that could be unexpressed, like we saw with the white flower. Those are traits that might disappear between generations. You'll notice recessive traits have a lowercase letter and a genotype. So for a recessive trait to appear, both parents have to have the recessive allele. They have to give the recessive allele to the offspring. So we see this in the picture. You can see with autosomal recessive disorders. So if you have two parents that are carriers for autosomal recessive disorders, then there's about a 1/4 chance that one of their children, will have the disease if that child inherited both recessive alleles. With autosomal dominant disorders, there's more like a 50-50 chance. If you have an affected parent and then an unaffected parent, there's still a 50-50 chance that the child would have the genetic disease because of this principle of dominance. If you watch the warm-up Ed puzzle activity, which I highly recommend, the peas are super cute, you would have learned that we inherit traits from our mom and dad. So we get one allele from mom and we get one allele from dad. So homozygous means the two alleles for the trait are the same, like big A, or little a. So big A would be homozygous dominant, little a would be homozygous recessive. Heterozygous means the two alleles for a trait, they're different, like big A, little a. So here you can see this compared in chromosomal structure. So you have homozygous, heterozygous, and homozygous, if that helps you visualize it a little bit better. So this takes us to Punnett squares, and a Punnett square is a chart used to determine possible genotypes of an offspring for a specific trait. So we are looking at the phenotype and we are predicting the genotype from the phenotype. So the inheritance of traits can be predicted using probability, and this just shows the probability of all of the genotypes. So we have dominant and recessive alleles, as you can see here. This is an example of parent 1 being heterozygous and parent 2 being heterozygous. When you cross two heterozygous parents, you always get the same ratio, the same genotypic and phenotypic ratio, and that ratio is 1/4 of the offspring would be predicted to be homozygous dominant, 1/4 of the offspring would be predicted to be homozygous recessive, and then 1/2 of the offspring would be predicted to be heterozygous. So each square signifies a 25% chance. So if the same genotype or letters show up in 2 boxes, that would be a 50% chance. If they showed up in 3 boxes, that would be a 75% chance. So it's important that you know how to display your knowledge of ratios in Punnett squares. So monohybrid crosses, it's just a cross that involves one pair of contrasting traits. So up to this point, you probably have experience with monohybrid crosses, but not dihybrid crosses. So today we're going to start with monohybrid crosses, and then I'll introduce dihybrid. A few sample problems. Red hair is dominant to white hair. So red hair would be displayed by a big R, white hair would be a little r. So big R would be red hair, little r would be white hair, big R, the heterozygous combination would be red hair because the big R, which is dominant, would mask the white hair trait. So you can see here these different crosses, and on the next slide, we are going to practice these a little bit together. So you probably remember doing this in 7th grade. So you set up the parental generation on the top and the side. So big R might be dad, little r might be mom, and then you can see the allele combinations. You kind of multiply them. So R times R, big R times little r produces big R, little r. And in this particular situation, every time you cross a homozygous dominant with a homozygous recessive, all of the offspring, 100% of the offspring is going to be heterozygous. In number 2, we cross a heterozygous parent with a homozygous recessive parent. And in this situation, we have 1/2 the offspring would be predicted to be heterozygous, and 1/2 the offspring would be predicted to be homozygous recessive. So take a second, and I'm going to pause. See if you can complete 3 and 4 on your own. Okay, so for 3, this would be your outcome. You would have 25% homozygous dominant, 25% homozygous recessive, and 50% heterozygous. And number 4, just to kind of show you how I'm getting this, the first box would be big R, the next box would be big R, little r, big R, big R, big R, little r. So we have 1/2 homozygous dominant and 1/2 heterozygous. The next sample question, short hair, which is represented by a big L, is dominant to long hair, which is a little l, in mice. What is the genotype and phenotype ratio of a heterozygous short-haired mouse crossed with a long-haired mouse? So before we even go to the next slide, we can kind of determine what we're crossing. So we have a heterozygous short-haired mouse, which would be big L, little l, and we're going to cross that with a long-haired mouse. So in order for it to be in order for it to have long hair, it has to be homozygous recessive with 2 little l. So this is what we're going to be crossing. So you can see that thought process at the top of the slide. Then if we make a Punnett square, we can cross these. And what we get is 1/2 big L, little l, 1/2 little l. That is the genotypic ratio because we're only dealing with letters. The phenotypic ratio only deals with physical appearance. So we would have 1/2 short hair, and that short hair comes from the heterozygous big L, little l. And then we'd have 1/2 long hair because we have 50% of that Punnett square is homozygous recessive, which means that would have long hair. We're going to do another practice problem. In goats, a recessive gene causes the goats to faint when they are startled. A farmer breeds 2 goats, and they have never fainted. Either goat has never fainted, and their first offspring faints 2 days after its birth. What must the parents' genotypes have been? Show the cross that resulted from this offspring. Okay, so the recessive gene is what causes the goats to faint. So we're going to make the letter for fainting be little f. So in order for a goat to faint, it has to be little f, little f. Big F is going to be our dominant allele. That means they don't faint. So let's make a few Punnett squares to try to figure this out. So both the parents, they've never fainted. So that means there's no way either of the parents can be big F, or sorry, little f, little f. There's no way they can be little f, because if they were, then they would faint. So let's think of the first scenario. What if both parents, since they've never fainted, what if they both were homozygous dominant? So if you see, before we even do the cross, if we cross 2 homozygous dominant parents, all of these boxes, 100% of these boxes are going to be big F, big F. And they had a child that fainted, so there's no way that's going to work. So there's no way that's going to work out. So what we need here is a heterozygous situation in order for this to work. So say one parent is homozygous dominant and the other one is heterozygous. That would still produce 100% probability that 100% of the offspring would not faint. That won't work as well. So what we need to do, both parents have to be heterozygous. So if both parents are heterozygous, we'll do a phenotypic ratio for this. So let's see where we have big Fs. 1, 2, 3 of the boxes have big Fs, so 75% would not faint. But there's the little f, there's the homozygous recessive. There's a 25% chance that they would have a goat with the genetic predisposition that would cause them to faint. So in order for them, 2 parents, to have a child with this recessive trait displaying in the phenotype, they would have to be heterozygous. Both parents would have to be heterozygous to donate this recessive allele to that goat. A dihybrid cross is more complex because you're comparing 2 traits, which means instead of 4 boxes in the Punnett square, you have 16 boxes. In this particular example, we're comparing eye color and height. So they crossed 2 heterozygous individuals. So both individuals are heterozygous and they have brown eyes and short height. So the dominant trait here is brown eyes and short height. The recessive traits are blue eyes and tall height. So we could come up with a ratio looking at each box. So to do a little bit of practice, if we interpret the genotypes, big B, little b, big S, little s. Well, you can see in this genotype, we have 2 dominant alleles, which means the eye color has to be brown and the height has to be short, because those dominant alleles mask the recessive trait. However, for little b, little s, the eye color would be blue and the height would be tall, because this is all homozygous recessive. All the traits are recessive. So when you're assessing dihybrid crosses, you want to always look at the genotype of one trait at a time to avoid confusion. And ratios are out of 16 instead of 4. So you would write them like 2 out of 16, 4 out of 16, 1 out of 16. So if we were to go to the previous Punnett square, we could hash out all of the phenotypic and genotypic ratios out of 16. So setting up a cross is a little more tricky for a dihybrid than for a monohybrid. So for dihybrid crosses, when we set them up, we use the FOIL method, which maybe you're having nightmares right now from your previous math class. I'm sure your math teacher taught you how to FOIL, but it's the way that you're able to figure out how to lay out the boxes. So here you can see the arrows, what they're matching up and how they figure out what goes on the top and then what goes on the sides of the boxes. So typically, if we were in school, normally we would do dihybrid crosses, but because we had to condense down the material, I'm just having you interpret them. And if you want to practice drawing them, just shoot me an email, and I have a couple activities that you could do to help you practice. In case you're interested in this, or you plan to go to AP Bio and you just want more information on it, I can send you more practice. So this slide explains the same thing as the graphic on the previous slide with how to use the FOIL method to come up with what traits to cross, what genotypes to cross. So I'm more interested that you can interpret a dihybrid cross than build one at this point. Although if you use the FOIL method, it's foolproof. You'll always get it right. In Mendel's experiment, they gave you a visual for this dihybrid cross, so you can see how each letter correlates. So in Mendel's experiment, yellow peas were dominant to green peas. So yellow is a big Y, green is a little y. And then round peas were dominant to wrinkled peas. So round peas are represented by a big R, wrinkled little r. So he crossed a yellow round pea with a green wrinkled pea. And then this was the result. 9 out of the 16 peas were yellow and round. 3 out of the 16 peas were green and round. 3 out of the 16 peas were yellow and wrinkled, and then 1 out of the 16 peas was green and wrinkled, homozygous, recessive for everything. And so you can see how he went from the P generation and then the F1 generation, and then that led him to the F2 generation. So I like this chart because it shows you what's dominant and what's recessive for things you guys have a connection to, like your shape of your face, and that actually says polygenic, so there might be more than one thing that affects that trait. And the same is for taste. There's more than one trait that affects your taste. So then the cleft in the chin, hairline, is it widow peak or is it straight? Do you have a hitchhiker's thumb? If you have a straight thumb, it's dominant. If you have a hitchhiker thumb, it's recessive. So to give you an example of that, I have a hitchhiker's thumb, and I'm not surprised that I do because my dad has it. And so my dad's recessive for that trait. My mom doesn't, but because my mom's probably heterozygous for that trait and my dad is homozygous recessive, I got the trait. So you can kind of see what recessive traits you have and what dominant traits you have.